So chapter 16, it covers development, stem cells, and cancer. We'll talk about the steps organisms take from a single cell to become a multicellular organism. We'll talk about epigenetics again, reproductive cloning, how stem cells work, and then we'll talk about the genetic basis for cancer and describe the multi-step path to cancer model. So for cells to go from a single cell zygote to different kinds of tissues and different organs and organ systems and all of that, there's several processes that have to happen. This requires cell division, growth, determination, differentiation, migration, apoptosis. So we'll talk about each of these coming up. But basically what's happening is that the genes that are expressed in the cell determine a cell structure and ultimately its function. So here's a little bit of a review. Match the following terms with their definitions. When it comes to these kinds of questions, I always like to start at the sentence and then go to the word. So which of these results in specialized cells? That's going to be differentiation. So 3 is A. And then which of these results in an increase in the number of cells? That will be cell division. So then we've got one left. So cell growth is going to result in cells with greater volume. So these next two terms are terms that students tend to get confused on. We've got cell determination and differentiation. So determination happens before differentiation occurs, and determination restricts the potentiality of a cell and commits it irreversibly to its final fate. So it's the molecular changes that cause a cell to become another kind of cell. But if we were looking at this under a light microscope, the cell would still look the same. It would just be expressing different genes. So then in cell differentiation, this happens when a cell is specialized in both structure and function. So when a tissue begins to express its tissue-specific proteins, that's when it's differentiated. And this is observable with a light microscope. So here's an example of determination and differentiation. So if we have an embryonic precursor cell and we look at the genes that are going on in that embryonic precursor cell, the MyoD gene is turned off and other muscle-specific genes are turned off as well. But if we look at a myoblast, which is a determined cell, microscopically it's going to look similar to that embryonic precursor cell. However, the MyoD gene is on, so we're beginning the process here. But the other muscle-specific genes are still off. Then, if we look at the muscle itself, which is a fully differentiated cell, that MyoD gene is on, and so are the other muscle-specific genes. So that brings us to stem cells. Stem cells are self-perpetuating and relatively undifferentiated, although there's different potentiality of cells. So a totipotent cell can produce all of the cell types in an organism. A pluripotent cell can produce any of the germ layers in an organism. What I mean by germ layers are there's either 3 germ layers or 2 germ layers depending on what kind of organism you are. So we have endoderm, ectoderm, and mesoderm in the middle, but some animals only have an endoderm and an ectoderm. Then a multipotent cell can produce many cell types, but it's limited, and then unipotent, uni is 1, right? So unipotent cells can make only one kind of cell. And as far as potentiality goes, in living organisms, this is permanent. So in vivo, it's permanent, but it can be reversed in vitro, which is it means in glass, but that just means in a lab setting. So you've likely heard of embryonic stem cells before. Many people have heard of embryonic stem cells before because of their potential for research. So embryonic stem cells are pluripotent, meaning they can make a number of different kinds of cells. And adult stem cells are either multipotent or unipotent. So they may be able to make several cell types, or they may be only able to make one cell type. So if we look at examples of stem cells, an adult would have stem cells in their bone marrow that can generate the different kinds of blood cells, but embryonic stem cells could differentiate into the different kind of blood cells or nerve cells or liver cells or so on and so forth. However, there was a pretty big breakthrough in 2007 where skin cells were transformed by viruses into pluripotent cells. So these were called induced pluripotent cells. So in a laboratory, they took skin cells and made them pluripotent like embryonic stem cells. And so that gives us stem cells that didn't come from, embryonic origin. So this is really important for research and that you understand this, because when people think stem cells, they think embryonic stem cells, but now we can have induced pluripotent stem cells, which are different. So let's go ahead and label the different kinds of cells. I think this picture is a little trickier than it honestly needs to be. If we look at the first cell, the blue cell here, let's see if I can get my pointer working. This blue cell here, this blue cell can divide to produce cells that turn into any of the different stem cells here. And then this next one can produce any of the 3 germ layers. And then the last row, like either the red one or the purple one or the green one, can produce only one of the types of cells. So if we look here, the blue one is going to be B, totipotent. The green one is going to be A, pluripotent. And then the red one or the purple one or the lighter green one would be multipotent cells. So that brings us to apoptosis or apoptosis. This is often referred to as programmed cell death. It's not a bad thing for these cells to die. It's really important for being able to get rid of cells that we don't need. So it can be used for pruning to create, fingers and toes, as is shown in the picture with the mouse paws on the right hand side. Or, to go from a tadpole to a frog, you have to prune that tail off. But this isn't just cells dying willy-nilly, it's a pretty regulated series of events. So when cells are dividing early on in the embryo, there's several things that are going to determine what kind of cells we get out of it. So cytoplasmic determinants are things like mRNA, proteins, and organelles that are unevenly distributed in unfertilized eggs. And so that already kind of sets the axis of the cell's body. And then induction is signals from nearby cell, like contact or growth factors. So that way the cells know where they are in relation to each other. So these things are helpful for the development of an early embryo. If we talk about migration, then I feel like people know migration when you think about like birds migrating or maybe monarch butterflies migrating, but cells can migrate as well. So there's different signaling events, again, like cytoplasmic determinants and inductive signals that orientate cells to an axis on an organism. These can also be chemical or mechanical signals, and the bundle of cells now knows which way is up and which way is down and which way is left and which way is right. This is pretty important. If you're thinking about a plant, the shoots and leaves need to be above the ground and the roots need to be below the ground. So now that the cells know which way they're going, they need to kind of form a pattern. So pattern formation is the development of spatial organization of tissues and organs. In animals, it begins with that establishment of the major axes. This requires positional information that tells the cell its location relative to the body axis and to neighboring cells. So cells can actually be reprogrammed and whole entire organisms can be cloned. So organismal cloning produces genetically identical copies of an organism. And, plants, it's relatively easy to do this. If you've ever taken a cutting of a plant, put it in water, got roots on it, you've cloned a plant. So congratulations. In the 1950s, they even cloned a carrot from a single carrot root cell, and that could grow normal adult plants. So plants are relatively easy to clone. Animals, on the other hand, are a whole lot trickier. So this requires removing the nucleus of an egg and replacing it with the nucleus from a differentiated cell. So in the 1950s and 70s, they've had some lucks with frogs on that. And in 1997, Dolly the sheep was the first sheep that was cloned, and obviously she was a mammal. But it's important to note that most nuclear transplants, including Dolly, don't develop normally to birth, and cloned animals often exhibit defects. So Dolly had lung disease that was really not common in young sheep. It was common in much older sheep, and she also had arthritis. So the extracellular matrix is all the stuff outside of the cell, and that's really important to functional organs and tissues. And the reason these things are exciting to look at and important to talk about in this unit is because it's not just the cells that are important. You have to have the extracellular matrix or the stuff around the cells. It's also to think about the applications of things like induced pluripotent stem cells and what they could be used to do. So let's review. During the process of bodily growth, organ development, and continuous cell division, there's a co-occurring destructive process. What is the term for that process? So that's apoptosis or apoptosis. Number 6, you love your dog and have a big pile of extra cash under your mattress. Why is cloning your dog probably a bad idea? So there's a whole lot of reasons why cloning your dog is probably not a great idea. With organismal cloning of mammals, we've seen that the resulting organism is not very healthy. And so if you loved your dog a whole lot, that would be a bummer. Also, the dog that you end up with might not be the same because it might have had different environmental experiences. So epigenetically, it could be different than your dog. And then here's another one to think about. If you love your dog and you love dogs in general, if you were to clone your dog, you would need cells from your dog inserted into eggs, and then would insert those eggs into a surrogate. So what conditions is that surrogate dog living in, right? I don't know. And then it's unlikely that they would just do one fertilized egg. So there would be a whole bunch of embryos of your dog clones implanted into this surrogate. And then, you know, do you get, like, what if you get 3 good ones, right? Do you take 3 of them home? So there's just a lot going on with that kind of makes cloning pets a pretty terrible idea, quite honestly. But anyhow, that was kind of ranty for that question. The big 2 would be epigenetics, so you might not even get what you thought you would get. And then also cloned organisms are not particularly healthy. So we've talked about cancer before. Cancer is a disease that's caused by uncontrolled division of abnormal cells in a part of the body. So there's actually oncogenes. Onco means tumor, so these are tumor genes that cause cancer. They do this by signaling for cell division when it's not necessary. And some viral genomes contain oncogenes, so they can actually insert rapid cell division signals into your genome. Proto-oncogenes then code for proteins that stimulate normal cell division and normal growth. However, a proto-oncogene could mutate into an oncogene, or a proto-oncogene could be amplified to overproduce a protein product, which could lead the cell cycle to come out of control. However, we've also got tumor suppressor genes, which are genes that inhibit cell division and prevent uncontrolled growth. So these guys help to repair DNA, they help to control cell adhesion, and they help to regulate the cell cycle. However, if these genes are mutated, then there's no tumor suppression, so that could lead to tumors. So here we've got a picture. In this first kind of column, if you will, there's a proto-oncogene, and let's say we have a translocation or a transposition, we get a new promoter, now we have an oncogene that produces normal growth stimulating protein in excess. Or we could have a proto-oncogene that was amplified, so now we have 3 copies of it. So instead of getting, you know, one little purple circle here, which is our growth stimulating protein, we get 3, so we have that in excess again. Or we could have a proto-oncogene that has a point mutation either within a control element, which can lead to overproduction of that growth stimulating protein, or we could have a point mutation within the gene itself, which causes us to produce a hyperactive or degradation resistant protein that stimulates for growth. So those are all things that can happen. So let's look at an example. P53 protein monitors the integrity of DNA before it's replicated. If the DNA is damaged, cell division is halted and repair enzymes are stimulated. And if the DNA damage is irreparable, the P53 protein signals for apoptosis. However, P53 is absent or damaged in many cancer cells. So, do you think the gene for P53 is an oncogene or a tumor suppressor gene? So the gene that codes for the P53 protein is a tumor suppressor gene. So with cancer, it's usually not one thing that's wrong, it's usually several things that go wrong. So researchers call this a multi-step model for cancer. Multiple mutations are often needed to produce the changes characteristic of cancer. So with this, usually there's at least one oncogene increasing the rate of cell division, and then there's the mutation or loss of several tumor suppressor genes. So lots of times these mutations are recessive, so they'd have to knock out both alleles for tumor suppression to stop tumor suppression. Cancer can run in families, as probably many of you know. Cancer runs in my own family pretty heavily. So researchers are looking for markers, like, for example, BRCA1 and BRCA2, which are tumor suppressor genes that are associated with breast cancer. And then tumor viruses can interfere with gene regulation and can integrate genetic material into the host cell. So HPV is probably the one that you're more familiar with. Not every strain of HPV causes cervical cancer, but there's an association between HPV and increased risk for cervical cancer. So that was chapter 16. We talked about the different steps involved in development, different kinds of stem cells. We talked a little bit more about epigenetics, and we talked about cancer.